Welcome all. We continue with the immunology module, and the topic for discussion today is antibodies or immunoglobulins. This is also very high topic. You will definitely get SBS from this particular topic. From the structure of the immunoglobulins, there'll be question. There'll be question from the type of antibodies and where it is present, etc. Always there is a question. Okay. So what is antibody? It is a glycoprotein which is produced by the plasma cells, that is the activated B cells. Okay, it binds specifically to antigen. It is located in the gamma globulin fraction on electrophoresis. So when you do the electrophoresis of the proteins, you will find that this they all will be present in the gamma globulin fraction. Okay, it constitutes around 20 to 25% of the serum proteins. Okay, what are the classes of immunoglobulins? There are different classes of immunoglobulins. They are IgG, IgA, IgM, IgD, and IgE. Okay, so these classes are depending upon the heavy chains. There are two types of chains, heavy chain and the light chain, out of which these heavy chains, they are on the basis of heavy chains, different classes are divided. Okay, if the heavy chain is of gamma type, it is called as IgG. If it is of alpha type, it is called as IgA. If it is a mu type, it is IgM. If it is a delta, then it is IgD. If it is epsilon, it is IgE. Okay. So what is the percentage of what is the specific key feature of these antibodies? So if IgG is the most abundant, so remember these features, they can be asked in the SBA. IgG is the most abundant type of antibody. IgA gives mucosal immunity. This is SBA. Then IgM is the first response antibody is the IgM. IgD is a B cell receptor. Okay, and IgE is responsible for the allergy and the parasites. Okay, so this is what is the structure of antibody. Okay, so these antibodies will have two parts. Okay, so this region, basically there are two types of chains, heavy chains and the light chain. So this is the heavy chain and this is the light chain. Okay, so the nature is a H2L2. Okay, so this is the light chain and this is the heavy chain which is there, and this has got two regions. This low region is the constant region, while this region is the variable region. Okay, so this variable region is of light chain and this is the constant region of the light chain. This is the variable region of the heavy chain. This is the constant portion of the this heavy chain. Okay, so this and this fragment is called as FC region, while this fragment is called as Fab region. This Fab region is responsible for the binding of with the antigen. Okay, so this fragment is responsible for binding of the antigen, while FC region is responsible mainly, this is the fragment crystallizable, but this is responsible for the further effects. Once the antigen antibody complex is formed, then further effects will be dependent on this. Okay, so these are the different parts or the structure of the antibody, basic structure of the antibody. So basically it is a Y-shaped molecule. There are four chains, two heavy chains and two light chains. So it is a H2L2 structure. So light chain is of other, again two types of light chains. Okay, so these light chains will be again of two types. Okay, so they may be kappa type or lambda type. Kappa type is 60% or lambda type is 40%, but one antibody will only have only one type of, either there will be kappa or there will be lambda. There will be no, both together will not be there. Okay, so light chain may be of kappa type or the lambda type. Okay. Then heavy chains. Heavy chains will determine the class of the antibody. We have just seen there are 5 classes, and these 5 classes are decided by the presence of these different types of heavy chains. Okay, that is gamma, alpha, mu, delta, and epsilon. Okay, so if you see this, there are sulfide bonds between these heavy chains. Okay, this is the light chain, this is the heavy chain, and this region is called as hinge region. Okay, so these heavy chains which are there, these heavy chains, again there are two types. In few types there this hinge region is present, and there is certain type in which there is no hinge region. Okay, so if you notice this hinge region is present in certain types, and there is no hinge region in the few types. Okay, and there are other, like here it is the variable portion and this is the constant portion. This constant portion is also here, it is CH1, CH2 and CH3. Okay, so few heavy chains will have CH1, CH2, CH3 conformation or formation, while few antibodies, heavy chains will have CH1, CH2, CH3 and CH4 also. Okay, so these are the two different things which are there. Okay, and this we have seen there is a Fab segment and there is a FC segment. This Fab is responsible for binding with the antigen, while this FC fragment is responsible for the biological activity. This we have already seen. Okay, so immunoglobulin with gamma, alpha, and delta heavy chains will have this hinge region, while mu and epsilon will not have the hinge chain. Okay, so mu and epsilon will not have the hinge portion or the hinge region. Okay, so let us see the difference between the variable portion and the constant region. We have seen that this is the variable region, this is the constant region. Of this constant region, in certain antibodies it will have be having CH1, CH2, CH3 configuration. Here few will have CH1, CH2, CH3, CH4 configuration. Okay, so variable versus constant region. Okay, variable region of heavy and a variable region of the light. Okay, so this will be variable portion of the light, this will be variable portion of the heavy. Okay, so and it is basically an antigen binding site. It contains hyper variable region. Okay, also called as CDRs. Okay, this is the hyper variable region and it forms the paratope. We have seen what is paratope. Paratope where they will identify the epitope. Okay, and that is the smallest unit of the antigen. So there will be paratope epitope complex formation. Okay, then coming to the constant region. Constant region will have the effector function. That means what type of response will be produced will be decided by this constant region. That is the effector function is there. Okay, it also determines the class of immunoglobulin, then complement binding and the placental transfer. This is the role of the constant region. Okay, then what are the domains? You have light chain and the heavy chain. Light chain will have variable light and the constant light portion. Variable light portion and the constant light portion. Okay, while heavy chain will have variable heavy part plus there will be CH1, CH2 and CH3, or there may be CH1, CH2, CH3, CH4. Okay, so heavy chain there may be little variation. Mu and epsilon will have four constant domains. Okay, constant domains will be four in mu and epsilon, that is CH1, CH2, CH3 and CH4. Okay, then the hinge region, this also we have seen between the heavy and light between the variable and constant portion there is a hinge region. Okay, but hinge region is present only in the IgG, IgA and IgD, while IgM and IgE won't have the hinge region. Function is it produces the flexibility and better antigen binding. This hinge region produces the better antigen binding and it is not present in IgE and IgM. Okay, you remember there is a immediate response by IgM antibodies. It is not there. Hinge region is not there. And in allergy, Ig type of antibodies are produced. So primary response and the allergic response antibodies will not have the hinge region, while other antibodies will have the hinge region. Okay, primary response antibody is a IgM. Anaphylaxis or the immediate hypersensitivity reaction is by IgE. These two will not have the hinge region. Just for the your remember purpose, you remember these two will not have the hinge region. Then there are two types of digestion. Okay, these antibodies will be digested by two mechanisms. One is called as papain digestion, another is called as pepsin digestion. Okay, remember these two names. There are two types of digestion. One is a papain digestion and another is a pepsin digestion. Okay, so if you see this is the pepsin digestion. What happens in the pepsin digestion? This antibody is broken over here and this FC fragment is lost, but this remains intact. Okay, so if you just imagine a person and the person is cut with the help of sword, what happens? The body is destroyed, but the upper part, the arms are together here. Both arms are together, but the body gets destroyed. Okay, so that is the pepsin digestion. In pepsin digestion, what happens? This Fc fragment is destroyed, but these Fab fragments remain together. Okay. What happens in papain digestion? If you see, the papain digestion is this way. So in papain digestion, these arms will get separated. Okay, but the Fc fragment will be present. Here the Fc fragment is preserved, but the arms are separated. Just remember papain. Okay, papain. So arms are separated in papain. Okay, pepsin, there is a Fc destruction. Pepsin, remember, for Fc, there is a, if this is destroyed, this remains together. That is pepsin. In papain, arms are separated. Okay, arms apart, they go apart. That is why papain. Okay, so these arms will go apart, but the Fc will be preserved. Okay, that is the pepsin papain digestion. Okay, then coming to the functions of immunoglobulin. So what is the function of immunoglobulin? First is a binding to the antigen. So once it binds to the antigen, it may cause neutralization of the antigen, it may cause agglutination, it may lead to the precipitation also. Then depending upon the site, this antigen binding site, there will be valency. IgG will have valency of 2, while IgM has got valency of 10. Okay, this you should remember. Okay, so antigen binding site is the valency. IgG will have valency of 2, while IgM is a pentamer antibody, so valency is a 10. Okay, then another function of antibody is the effector function. Okay, effector function is the, this Fc fragment is responsible for the effector function. So once this antigen antibody complex is formed, after that there may be complement activation, there can be opsonization, there can be ADCC, that is antibody dependent cellular cytotoxicity, and second, there can be placental transfer. This is a feature of IgG. Okay, so another function of antibody is effector function through Fc part. Okay, this is what will be the effector function? It may be complement activation, there can be opsonization, there can be antibody dependent cytocellular toxicity or placental transfer. Okay,